Today we're doing an earthworm dissection, and before we actually get down to dissecting the earthworm, I want to take a minute and go over some anatomy. We're going to start with the digestive tract, which is in green here. In the beginning of the digestive tract is the mouth, which is at the most anterior end of the earthworm. From there we're going to go into our first dilated structure here called the pharynx. The pharynx is an organ that actually pulls food into the earthworm. That's its job, is to actually vacuum or pull food, soil, into the earthworm. From there the soil passes through this long green tube. In real life it's not green, but this long green tube that I'm pointing to anyhow, until it gets to the crop. The crop is a pouch, a very thin pouch, sort of like a balloon, and it's designed to increase in size because that's where the soil is going to get stored until it makes it to the next phase of digestion called the gizzard. The gizzard is a very muscular organ, so if you push on it with a probe, you'll feel the resistance of all that muscle. And it's muscular because it digests and grinds the food. So the gizzard is going to do two things. It's going to release digestive enzymes, and it's going to grind the food up. It's going to mix the soil up with the small bits of organic material that this earthworm wants to digest. From there, we're going to go into the last part of the digestive tract, referred to as the intestine. Now the intestine is probably going to make up 70–80% of the earthworm. And it's going to be that way for a reason, because we're trying to digest and absorb small bits of organic material mixed in with lots and lots of topsoil. So in order to do that, we have to create lots of surface area. And we're going to do that in two ways. One, we're going to have a very long intestine. And number two, if we look at a cross section of the intestine, you'll see that right here we have an infolding of the intestine, and it forms a loop. And this loop is going to greatly increase the surface area in the intestine. You can see that if the tissue just came straight across, we would lose all that additional surface area for absorption. So this structure has a name. It's called the typhlosoal, and its job is to increase the surface area for absorption. Now let's take a look at the blood vessels. We're going to see three major blood vessels on this worm. The one blood vessel you can see right here is probably going to be the most prominent. It's going to be called the dorsal blood vessel, and its job is to return blood to the headward region. And then we can see some dilated blood vessels here on either side, and these dilated blood vessels are called hearts or aortic arches. And their job, they've been modified to pump blood. They are modified blood vessels, and that's exactly what a human heart is. It's a modified blood vessel, except it's become more modified and more specialized than the heart of an earthworm. Usually you'll have about 5 sets of these hearts, and if you're lucky you'll see 2 or 3 on an actual dissection. If I turn this over, on the underside, the belly side of the earthworm, you'll see another blood vessel. And this is called the ventral blood vessel because it's on the ventral aspect. And this blood vessel actually returns blood back to the body of the earthworm. Along with that blood vessel runs a nerve. And you can see that nerve right here. It's a very large nerve, and we're going to call it the nerve cord. It's sort of analogous to your spinal cord. And that nerve cord is going to run the length of the earthworm. Now at the most anterior aspect of the earthworm, up here, this nerve cord is going to form a large, at least with respect to the earthworm, a large focus of nerve tissue where nerve impulses actually get processed. This is called the brain right here. This is the brain. It's going to be very small in the earthworm, but if we see it, I'll point that out to you. Now let me go ahead and put this back together again. And you can see 2 cream-colored organs on either side. These cream-colored organs are going to be called the seminal vesicles. And their job is to produce sperm that will be used to fertilize the eggs of another earthworm. And down here we have smaller structures, going to be similar in color, maybe a little bit whiter, but similar in color. And those are going to be your seminal receptacles. They call them receptacles because their job is to receive something. they actually receive the sperm of another earthworm. And they store that sperm temporarily until the earthworm is ready to fertilize its own eggs. Now, keeping with that thought, we've got another large prominent structure here on the external surface of the earthworm. You can see this band of tissue right here, and this is called the clitellum. And its job is actually to secrete like a viscous material that will eventually become the cocoon. And inside the cocoon will be the fertilized eggs. Now, also noted here, you can see these large flaps of skin right here. These are called septa, and they actually separate the internal part of the worm into compartments. So they are responsible for segmentation. The earthworm is not only segmented externally, but it's segmented internally also. And in each one of these compartments are different organs. Now, the actual compartment here, all this space between the wall of the earthworm and the intestine, or the digestive tract of the earthworm, is referred to as the coelom. And the coelom is broken up into segments by these septa. All right, let's go ahead and actually start the earthworm dissection. So we have our earthworm in the dissecting tray, and these are fairly large earthworms. So what I would like you to do is to pin the earthworm diagonally across the dissecting tray. And we're going to take 2 pins, and I'm going to take the one pin and put it in the anterior aspect. Now, the anterior aspect is going to be the larger part of the worm. This is the head portion of the earthworm, where all the organs are going to be located. And we have to put the dark side up. Now, the dark side is very pigmented, and that is the side that needs to be up in order to see the organs. So I'm going to go ahead and put a pin straight in the anterior aspect of that earthworm, and then I'm going to take another pin and put it straight in the posterior aspect of the earthworm, just like that. And again, we want the dark side up. This is the dorsal side of the earthworm. The underside, or belly portion of the earthworm, is the ventral side of the earthworm. So now we're going to go ahead and get to our dissecting. So we've got our scalpel. Note the clitellum right here. I want you to begin your dissection posterior. So anterior is headward, posterior is towards the anus. I want you to start posterior to the clitellum, because the only structures in here are essentially the intestine. So this is where I want you to practice. And if you make a mistake, it's not going to be too terrible. So we're going to start dissecting there. So you pick up your scalpel, you're going to hold it like a pencil. And I like to use the very tip of the scalpel. So I hold it almost vertically. And I'm going to hold it at an angle also. I'm going to grab my forceps and I'm going to begin cutting. Now when I cut, I always cut away from my hands or between my hands, but never toward. You never want to cut towards your hand. So I'm going to cut away from my fingers. And I'm going to start right in the midline. And I'm going to go in very shallow. And I'm only going to cut maybe a ½ an inch to an inch at a time. Very shallow. And then I'm going to take my forceps and I'm going to try to open that up. That cut I just created, I'm going to try to open it up a little bit. I'm going to pull back the skin. And I'm going to go ahead and I'm going to pin it. So I'm going to put the pins in probably at around 30 degrees or less. And that way I've got some room to not only see, but to work in here. So I've pinned one side of the skin, and I'm going to turn the earthworm just a little bit, and I'm going to pull that skin back. I'm going to sort of tent it up. And you can see the septa start to appear. And I'm going to cut those septa because that's going to release the skin. And I'm going to be very careful is to not cut the intestine. If you cut the intestine, you're going to see some black material ooze out, and that is simply topsoil. That's all it is. That's a signal or an indication that you have cut too deep. That's about ½ an inch or so. Now I'm going to take the remaining skin and I'm going to pull it away just a little bit, and again I'm going to continue to cut very gently, maybe ½ an inch to an inch at a time. And I pull back the skin, or reflect the skin as we say in biology, and I put another pin in at about 30 degrees. and I just continue to work my way down towards the anus. So again, I'm going to go ahead and do the other side, working about a half an inch to an inch at a time. And I'm going to go ahead and pin this side. Now, as we do this, if you're working in a group of two, make sure that only one pair of hands are in here at a time. So I'm going to go ahead and go down the other side here. And again, I just work my way down a half an inch to an inch at a time. And we can see this dark structure begin to appear on top here. This dark structure right here is the dorsal blood vessel. That's the dorsal blood vessel. The space between the skin and the intestine, this is the intestine, is going to be called the cecum. And that cecum, coelum, that coelum is our space, is our body cavity. And that body cavity, or coelum, is going to be divided into compartments by the septa I was cutting through. Now, if you happen to cut too deep, and I'm just going to go ahead and show you what's going to happen. I'm going to cut a little too deep here, and you can see the soil begin to ooze out. That's an indication that you have cut too deep. You need to back off and go a little bit more shallow. Now, I'd like you to practice and proceed all the way down to the anus. After you've gotten there, you've gotten in some practice, then go ahead and turn the earthworm around, and let's go ahead and begin working towards the anterior aspect of the earthworm. And what you're going to do is you're just going to cut right through that clitellum as though it weren't there. So I'm going to go ahead and again, cutting very shallow, about a half an inch to an inch at a time, I'm going to cut right through that clitellum. And I'm going to, with my forceps, I'm going to pull back the skin very gently. You can see some of the septa are still intact there. I'm going to go ahead and I'm going to cut through those septa. I'm going to pin this back, making sure to keep my pins very shallow or coming in at a very low angle. Now I'm going to go ahead and do the other side. I'm going to take that skin, pull it back, and cut those septa. And I'm going to work my way anterior. Now eventually, as I get close to the mouth, what I want to do is I want to remove that pin so that I don't bump into that. I'm going to go ahead and I'm going to turn the pan again, and I just go back and forth until I complete this. So I'm going to reflect the skin a little bit here and cut those septa. Go ahead and I'm going to pin this. And I'm going to continue my way towards the anterior. Now you can see right here this saran wrap-like material beginning to appear. I got a pin right underneath a little bit of it here. And this saran wrap-like material, you can see it right there, is simply the cuticle. And that cuticle is going to be responsible for protecting the earthworm, both in terms of dehydration a little bit and in terms of protecting it against abrasion as it works its way through the soil. Let's go ahead and go a little bit further here. I'm going to tent the skin up a little bit and continue cutting. And I'm going to try to be very careful that I don't puncture any of the organs here. There's a lot of good organs here and we don't want to cut them. And we're almost to the mouth. We cut through some remaining septa there. And then one more cut should do it. So the skin's going to be adhering very tight here, so I've got to be careful. The brain's up here too, and if we're lucky, we'll get to see the brain. And there we are. We've dissected our earthworm, and now all that remains to do is to go over some of the anatomical parts. And the first thing I want to show you here, in case it gets destroyed, you can see these white structures right here. I'm going to try to hold this down just a little bit. Maybe we can get a close-up shot of this. And you can see the two little white structures right there at the tip of the pin. Those two white structures are the brain. Eventually the nerve cord ends in those two white structures. That is the brain of the earthworm there, not quite as big as yours. Now let's go through the different parts here. The mouth is the very opening at the very anterior tip. And then the next part of the digestive tract is going to be this part right here. This dilated part right here. That's going to be the pharynx. And then underneath here, this is not going to be possible to see, but underneath this material here is going to be our esophagus. And then we go into this pouchy area. You can see how soft that is. You want to push on that with the probe a little bit. Part of dissecting is not just naming the parts, but being familiar with what the parts look like, what they feel like. And this should be very soft, very distensible, so that it can enlarge easily. If this was thick material, it would not be able to enlarge. That's going to be the crop. And then you have this structure right here. Notice how firm this is. Very muscular, very firm. This is going to be our gizzard, where the food gets ground up. From there, we're going to go into the long intestine. And again, we have this long intestine to create lots of surface area. Traveling along the top of the intestine is our dorsal blood vessel. You can see that dark structure right there along the dorsal part of the worm. This is all going to be our coelom, and you can see the various septa along there too. Now let's go ahead and take a look at these cream-colored structures right here. We're going to have to actually move them out of the way in order to see our aortic arches. So as you're dissecting, just give them a gentle push. Try not to tear them, but just kind of push them out of the way a little bit. Those are our seminal vesicles. And again, those are used to actually produce the sperm. I'm going to move those seminal vesicles out of the way a little bit. Those were the seminal vesicles. Anterior to the seminal vesicles are some smaller structures. You can see it right here, and you can see one right there. And on the other side, they are not as good, maybe down in here a little bit. Those are going to be our seminal receptacles. Those receive the sperm of another worm and store it temporarily until fertilization is ready to take place. Now, what we can do here, I'm actually going to, I see a little bit of skin still left on here. I'm going to pull it up just a little bit and cut that skin away very carefully. There's some good structures there, and we don't want to injure those structures. And what I'm looking for now, after I move the seminal vesicles and the seminal receptacles out of the way, is I'm looking for those aortic arches or hearts. And they're just going to be small black threads. Don't expect a lot. And you can see one right there. If we can zoom in on that right there, that is a very nice aortic arch right there. That's a very nice heart. That's about as good as it's going to get. You can see one on the other side also. If I can tease that out, there it is right there. Another very nice aortic arch. And you can see another one here, another one here. And when you tease these, kind of tease across the worm like this. You don't want to go this way because you'll just rip them in two. So you want to kind of go this way. And maybe if you can get underneath one, lift it up just a little bit, just as I did there. You can see another one come into view. That's all you're going to get. Now, if you can get 2 or 3 of those, you're doing very good. You're doing very well. And there should be around 5 altogether. Sometimes a little bit more, sometimes a little bit less. There is no perfect worm. Now, the last thing we need to do is we need to roll this intestinal tract, this digestive tract, out of the way a little bit. I don't just want to cut it up, but I want to roll it out of the way. So I'm going to take my forceps and kind of pull it over just a little bit, and I'm going to cut some of the septa that are holding it in place. Again, keep doing that very gently, and I want to do this towards the anterior of the worm because this is where my largest structures are going to be. And what I'm looking for here is I'm looking for the nerve cord. I'm looking for a white fiber. It looks like a white thread. And I just very gently keep pushing it over, cutting the septa that are holding it in place. And where is it? It's coming into view right now. So I'm going to move this over just a little bit. I'm going to hold this out of the way so you can see this. And if we look in there, you can see this white structure right there. This white, looks like a white piece of thread. And that white piece of thread is going to be our nerve cord. You can see it right there. And then along with it, you can see this very small black fiber running along with it. You can see it right there. You can see it right in through here. Try to clean that up just a little bit. That is going to be the ventral blood vessel. And that's going to run with the nerve cord. And you should know that. And that's going to be it for the earthworm dissection.